Never was an early riser, used to be an up all nighter. Never saw the morning light quite like I do now. Never said no to a party, never started saving money. But everything is different since you've been.
Hi, so the second type of macromolecules we get to frequently encounter with in our life, inside of our life, the cell, is lipids. One very distinctive, which distinguishes itself among the other types of macromolecules. The distinctive feature of the lipids are hydrophobic. So, like, unlike carbohydrates, which is basically polar nature, so that they are hydrophilic, but all lipids are hydrophobic, with the slight exception of phospholipid that we are going to take a look at later on in this section. So since it is a hydrophobic, lipids are all insoluble and also hydrophobic. Okay. They don't really want to have anything to do with the association with the water, because the water is polar molecule. Why? Like in the case of carbohydrate, we determine why carbohydrates basically would be hydrophilic, because of the presence of this oxygen. Many abundant appearance of oxygen will inevitably make the carbohydrate, which is a kind of a charge distribution, is unequal, so then they will become hydrophilic, polar. Quite the opposite you can see in the case of lipids, so that you don't really have any oxygen, not too many, very little oxygen so to speak, sequestered in only one side, and the vast majority of other areas, all you have is not too many elements of having potential of the polar, but simply carbon and hydrogen, carbon and hydrogen, carbon and hydrogen. So then it will naturally make those areas very hydrophobic, non-polar. So there are several different types of different subgroups of lipids exist, including first fat. So here usually we have the fats, which is solid in room temperature. In everyday, it's like we call this an ambient temperature. Fats usually exist in the form of some kind of solid. On the other hand, oils is a liquid form on the same temperature. But whether it's a fats or oils, they are actually the same type of fats. Okay. The only difference is they are a slight structural component. The structural component, the nature, the chemical nature is slightly different, and then that makes this type of differences, whether it's a solid or a liquid, at a certain range of temperature. By the way, all fats are actually made of the monomeric unit making these fats is called fatty acids. Okay. So these fatty acids, certain number of fatty acids, have a maximum, actually 3 fatty acids, can have a specific condensation reaction with a certain type of molecule called glycerol. It is a type of alcohol having 3 room for this alcoholic properties. This glycerol, to us, glycerol is probably rather well known. It's greasy the texture, but anyway, glycerol is a type of alcohol. And then with this combination of the fatty acid and glycerol, had to have that particular condensation reaction, and then generate a compound we recognize as triglyceride. Its name is the triglyceride. I think in the version of the lecture handout, there is some kind of typo misspelled, and so I want you to correct into this form. So fatty acid plus glycerol make up a triglyceride, and there are many different. So these are all so many different types of fats and oils do exist. However, all of them are basically one or the other form of types of triglyceride. Although to us, all these triglycerides are known as this, right? But these are simply this, because almost all of those fats that we obtain from our food source are actually in this triglyceride form, and that's why, especially in medicine, they are specifically kind of recognized as this particular, whatever you call this, neutral form of fat. I don't really like the name, but this is the triglyceride is the one that you knew as this. So structurally, the triglycerides is made like this. So this is one unit of such a fatty acid. So 3 of these fatty acids are attached. So this is since this is a condensation reaction, 3 molecules of water will be extracted, and then now you have one fat molecule. And that is the reason why earlier in the previous section, that when I was toying with the the picture of these 4 comedians, then I just compared. That's the reason why I compared the shortest guy to the fat, the contrary to the general expectation, because as you can see over here, fats is the one of the major example of lipids, and from what it's we can take, the fat is probably the smallest the macromolecules among those 4 different types of macromolecules for this reason. So this is the maximum. This is all you can build as just a fat molecule. But in the case of carbohydrates or nucleic acid or protein, you can build huge, much larger and larger form. You can expand. But here in the case of fat, no more. Another example of having this. By the way, this particular type of the condensation reaction, which is a covalent bond, is called ester bond, which distinguish, the ester bonds are the covalent bond, the condensation reaction bonds to assemble these fats from the fatty acid, individual monomer, which is a fatty acid. But here there are 2 different kinds of fatty acids, and due to that difference, actually the fats generated as a result of this assembly of individual fatty acid can also be divided into 2 different types. One is called saturated fat, and the other one is called unsaturated fat. It's all is dependent upon how many carbon double bonds in their fatty acid chain. Here, this is some example of the different types of fatty acids. All fatty acids are at one end we usually distinguished as a head, because you have this particular chemical group, which is a carboxylic group, have some oxygen, so it may have a slight polar chemical characteristics, but other than that, all the other areas having nothing but carbon and hydrogens, which make these whole fatty acids are extremely hydrophobic. So we usually designate them as a hydrophobic tail. Hydrophobic tail and hydrophilic or slightly little hydrophilic head. But that is not the actually point that we are going to look at. Some fatty acid, if you examine each individual carbon in terms of their maximum capacity of forming the covalent bond, each carbon is exhausted. Each one is like all the maximum capacity is used up with the hydrogen and or the neighboring carbon. But on the other hand, some cases you do have this double bond. So like instead of having double bond between these two neighboring carbon, you could have another hydrogen, like covalent bond with hydrogen, but these guys rather does not want to do that. So that's the reason why this particular type of a fatty acid is called unsaturated. Okay. Whereas this type of fatty acid is called saturated, but these two are unsaturated. By the way, actually these two type of the fats happen to be one of those famous unsaturated fatty acids, like omega-3 and omega-6 fatty acids, which are well known to us by like their health beneficiary effects. So those are the difference between saturated and unsaturated fats. But among the two different among the unsaturated fat, there are actually we can distinguish two different types of unsaturated fatty acid. One is here, as you can see over here. This is a conventional natural form of fatty acid. All the natural form of fatty acid, unsaturated fatty acids, are in this configuration. We distinguish them as a cis fatty acid. However, artificially, we can also sometimes generate the other configuration of this double bond. We now call it as a trans fatty acid. What's the difference? As you closely look at this, the position where the carbon double bonds are made, and then look up at the position of the hydrogen and the carbon. So like in the case of a cis double bond, between these cis, one is hydrogen is up and the other, probably the carbon, is running, and this would be the relative orientation of the carbon and the neighboring carbon and the hydrogen around this double bond. And then we call this as a cis configuration of this double bond. Because why they are called the cis? The hydrogens are in one side and the carbon is on other side. They are clearly separated, so they are arranged in the same side. So that's why the cis meaning the same, so cis double bond. Okay. On the other hand, so like this cis configuration, like you are just like put your hands up all together kind of a configuration, is a cis double bond. On the other hand, you can have the other alternative configuration of trans configuration where you have this. So like they were an alternate side of, just like you are in this disco mode configuration. So this type of double bond configuration, we distinguish it as a transformation. So you can have like alternatives of whether you form this cis double bond or trans double bond as unsaturated fatty acid. But interestingly, all naturally occurring unsaturated fats are all cis configuration. And the thing is, when you have this cis configuration, as you can see in this structure, you have this slight kink in the angle because of this angle generated by these two carbons forming this double bond. So if you only have this cis configuration of a double bond, and then you naturally have your fatty acid chain will have some kind of skewed angle at one direction only. And like I said, all the naturally occurring unsaturated fats have this cis double bond configuration, so that fatty as a one fatty acid, if you are a fatty acid and then unsaturated, then your tail, the fatty acid tail, depending on how many double bonds you have, will have this type of a kinked angle, as opposed to if you are saturated fat, then you will have this type of linear, all nice linear arrangement, or the straight line configuration. So as a consequence, if you have many unsaturated fatty acid, especially having more number of double bonds, then with the same number of this fat, the volume it takes up all those different fatty acids in one limited space will become larger. In other words, all individual components within that area will be more flexible. So that's why these all unsaturated fatty acids will be liquid, whereas this saturated fatty acid will become solid. So that's what makes the difference between oil and fat. So like fat, like butter, which is a solid room temperature, is because it's mainly exclusively consists of saturated fatty acid. So they are solid because they are tightly compact, organized. And on the other hand, this liquid form is because their fatty acids are unsaturated, so they are moving around more freely because of this angle of individual fatty acid. So that's why they behave as an oily, liquid-like. And all these are the plants. Plants make a lot of those unsaturated fatty acids, so that's why a lot of those oils, like the vegetable oils, as you can easily imagine, are all from the plant source. Okay, so this saturated fat and unsaturated fat, and then let's take care of the business of this trans fat, because trans fat, we are all we have all heard that trans fat is even worse than the saturated fat. So how did you make this trans fat? You have this saturated fat like this, solid, and you have this typical unsaturated fat, which will become liquid. Now, this, everyone knows that saturated fat is which is not healthy, because it will cause more health hazard, because it is a solid. So if you just consume too much of those saturated fat, it will in your blood vessels, in your circulatory system, these solid fat will cause more of those the clogging problem. So that's one of the reasons why you want to avoid those saturated fat consumption. So yeah, so in real world, I mean in perfect world, you'd rather actually have only the unsaturated fat consumption, which will be much healthier. But in reality, you cannot, because a lot of times actually very inconvenient. So like imagine butter. I love butter. The only thing that I can complain is like too solid, so I have to wait for a while to have this manageable the texture before I apply them on my bread. But other than that, I really love butter. How about the margarine then? Margarine is something like a fake butter made from originally from vegetable oil. How do they make the margarine? So that they can have this butter-like oil properties out of the oil. So they generate some kind of a chemical conversion, which is called the hydrogenation. So this is artificially you are trying to insert the hydrogen among different side of this randomly among different side of this double bond area, so that they can eliminate reduce the number of double bond. Then if you do so, then it will be more like a more saturated fat. So that's how you generate this margarine, okay, which is actually in at least in terms of the texture, which is more amenable, so that that's the better part. But still the taste doesn't taste as good as the butter, so I don't really like margarine. But the problem is if you just generate such the margarine, then you inadvertently get to generate the trans fat, because in human hand there is no way you can control whether that the remaining double bond somehow in the trans configuration or cis configuration. So it will be a mixture of. So some of the hydrogen and the double bond configuration will be the trans position, which was not natural. In nature, you have never will see such things. So these trans fats are unnatural and better yet, worse yet, it's we all know that it is actually more serious health hazard than the saturated fat. These are the difference between cis and trans fat, as I already have demonstrated this. So this, whether this or that. Okay. So, and as a result of the hydrogenation, they were generated like that. And so many other different examples of such trans fats you can see in our everyday life. So these unsaturated fats are like due to this angle, this is an oily, but the trans fats, because of this random trans and cis, it can have very like saturated fat, the likeish things. But the question is why this trans fat is such a health hazard? Is that simply because of this, like more like this, the fats like a texture, so that it can become also more solid inside of our body to cause a similar the problem as those saturated fat do? Maybe to some degree, but more serious problem of this is also like it's very similar to the case of that artificial sweetener, the sucralose. Why the sucralose is unmetabolizable? Because we have never seen such thing like that. We have no time to develop any enzyme to deal with that specifically. So it's the same thing for this the trans fat too. We don't have any enzyme to efficiently break down this trans fat. So the two most major area of causing the damage from this trans fat is, one is the liver, and the other one is our blood vessel. Why? Because in the liver is the area where the breakdown of all different kinds of fats are made. Then the liver, if you were liver, and just a totally new stranger came in and then asking for, begging for the digestion, please destroy me, but liver don't know how to, because they don't have any tool to cut them. So it will just sit there causing some of the problem. Probably that's what's going on. And blood vessel, all this undigested, big chunky trans fat is running through, so it will definitely cause, eventually has a lot of those clogging related problems. So that's probably those two major reasons why these trans fats are such a big serious problem. Once again, these are all related with in the case of this sucralose, artificial sweetener, and in the case of this artificial fat, something like a trans fat, all are something that are highly associated with, specifically associated with the enzyme specificity. All those compound materials, whether it is a synthesis, whether it is a breakdown, destruction, all must be dealt by specific enzyme. That's how we run the business inside of our cells. And if you don't have any such specializing the professional to deal with, then you are in trouble. Is once again the lessons we can the takeaway we can have out of these two different occasions of the stories. Now, phospholipids. It is something different, slightly different from fats, but actually it is something that is highly related with fats. The difference between phospholipid and the conventional fats are here. Unlike the conventional fat, where 3 fatty acids are attached. Now the number of fatty acids attached, you get to see only 2. So one is missing. Why? Because there are something in glycerol. Remember this glycerol having 3. Glycerol is a kind of a plug-in module. We don't really have to worry about the structure of the glycerol, but just imagine this glycerol is a 3 plug-in module, each of which can have this all fatty acid chain can be attached. Now imagine you don't have this thing anymore. It's not available for the third fatty acids to be inserted. Then you only deal with those 2 of them. Why this the third unit of the OH is not available? Because this OH unit is playing with somebody else at the other side of this whole unit. It's what this phospholipid has. So these guys are playing with some other very distinct, like the other side of the spectrum of this molecule, which is extremely hydrophilic, which is a phosphate. So this is what, if you actually look up just once and for all, here you don't really have to worry about any actual chemical structure of this phospholipid, but just once and for all, if you look at this glycerol unit the plug-in unit is now acting to connect the other side with phosphate, which is extremely hydrophilic because of this oxygen. And better yet, once you have this phosphate, and phosphate will bring in the other his or her friends, which is also hydrophilic. So you will have a highly expanded area that can act now as a hydrophilic. So this two side things. One area is a hydrophilic and the other area is a hydrophobic kind of property has been way more exaggerated in phospholipid because of this structural arrangement. So having you just seen this, then now you just forget about everything about this. Now as a student in biology, all you gotta worry is this is the preferred form of viewing individual phospholipid in biology. Yeah, so just perfectly fine. What's wrong? You have this hydrophilic head, which is mainly made by the phosphate group. Phosphate and other friends generate this hydrophilic head, and hydrophobic, the fatty acid chain, hydrophobic, the tail. So that's the two area of this two distinct in terms of its the hydrophilicity or hydrophobicity, the charge. So we described such kind of a two-side property as an amphiphilic or amphipathic sometimes, which of which is simply indicating just two sides of this charged and uncharged area. Okay, so if you have this kind of the molecule, and let's say you spread tens of thousands of this phospholipid molecule in the water, then what's going to happen? It will assemble in two layers spontaneously in the water, and then it will be the basis of the assembly or establishment of a cell membrane. That's how we that's what, at least what the biologists believe. Like that's what just happened in the beginning to have a first type of cell. In order to make cell, you need at least have this first form of the cell membrane, and also inside of which you better have this genetic material, as I went over in the first lecture series. And this the chemical property of these phospholipids now, for that matter, must have played such an extreme role for such formation. So this figure illustrates such feature. So as an individual phospholipid, if you have so many such phospholipid molecules, then they all together here, what's going on over here? They aggregate together. The hydrophobic part of this phospholipid will aggregate together. We have seen such thing like that, and then remember hydrophobic interaction, right? They will get together. Why? To avoid the contact with the water. But here, good thing about this phospholipid is they also contain this hydrophilic surface area, which has no problem in dealing with the water. So it is really ideally they can easily form this type of arrangement of two layers, which is called the phospholipid bilayer. That is the structural motif of the cell membrane. So you can have an extended phospholipid bilayer. Eventually they will have this closed structure like that, and this is the beginning of cell. So outside the water, inside of water you have this cell membrane. Is such a structure, like bigger form of such a phospholipid bilayer acting as a kind of a cell membrane like that. So
Cellular membranes are made of a double layer of lipids, or lipid bilayer, in which parallel layers of lipids face away from each other. In this case, the lipids are phospholipids, with two fatty acid chains, or tails, attached to a phosphate-containing head group, hence the term phospholipid. The head groups are hydrophilic, meaning water-loving. They form weak bonds with the surrounding water. The fatty acid chains are hydrophobic, meaning water-fearing. They do not dissolve in water and are actually excluded from water because of the weak attraction of water molecules for each other. The hydrophilic and hydrophobic characteristics of the phospholipids keep the membrane intact without requiring any energy on the part of the cell. The head and tail groups in membrane lipids are the right size to line up side by side, forming the sheets of membrane that surround the cell and divide its interior into compartments.
Okay. So depending on how many phospholipids are available at a certain area, you could have this like just bilayer sheet like that, or a little bigger structure called a liposome, or some other times when you have only a small number of available such a phospholipid, then you can also have instead of having this bilayer, you can also have this type of mini scale structure we now distinguish as a micelle. So all of this just some particular form of this spontaneous structural form generated from the individual phospholipid membrane, especially the liposome. In the case of this liposome, which we can artificially easily generate in the laboratory, has such a tremendous the usage, utility, application, especially in these days. For example, a lot of times in the pharmaceutical company, when they develop a new drug, to illustrate that, actually we have to first understand this each of these different structure of the basically the similar type of molecule, they can easily fuse one another because they are same type of molecule. So easily, if this is a one small part of a cell membrane, let's imagine that this is a part of a cell membrane, then this liposome can easily fused with and then integrated as a one, just like a vapor, like a liquid vapor can be fused and combined together easily and then form one big bubble like structure. So are these different types of such lipid structure, phospholipid structures to be exact. So that's the one of the advantage that is being taken actually by the cell tool, which we will be able to see later in other chapters when we look at the trends of the molecule inside of the cells actually occurring. But here, this liposome, the point of the matter is it can be easily be fused with a part of the cell membrane. What is the significance of such a property? That once you put some of the delivery cargo inside of this liposome, it will be much easier for this liposome to be delivered into the actually inside of the cell. Because once they have fused, then internal content will be dumped easily into the internal space of the cell. So that's why all those pharmaceutical company, when they have developed the drug, especially if the drug is something very hydrophilic, if they are very hydrophilic, then it will have a difficulty in penetrating cell membrane, because cell membrane, because of this huge thick layer of hydrophobic part, they will not be able to take any polar molecule easily. So a lot of such hydrophilic molecule, including this DNA, RNA, and those. In the case of this vaccine, DNA and RNA vaccines are in these like RNA vaccines from Moderna and Pfizer, or like DNA vaccines from the AstraZeneca, are all delivered inside of this liposome so that they can be more effectively go into our cells internal space. Is how they do. And also they have all kind of other tricks. So on the surface they have some of the specific recognition markers, attachment anchor, and you name it. You can have many different like variety, the variation to make this liposome more effective in specifically targeting to any particular part of your body. Is how make use of this liposome in this case. The next example of lipids is cholesterol. Even though there are several other different types of lipids do exist, but we cannot go over all of them. So this is easily known as the precursor of a steroid hormone, which means if you want to synthesize in your body many different types of steroid hormone, two of such famous steroid hormone that perhaps you are already aware of is estrogen and testosterone. But in addition to that, we have so many other different types of steroid hormone. And the cholesterol in our body as a multicellular organism, although we individual cells actually is capable of also synthesizing cholesterol, but mostly they are made in bulk in the liver. And later actually you will have a problem of this delivering this cholesterol through your bloodstream, but that is not something that an issue that we need to worry about here. So let's just focus only the chemical individual chemical aspect of this cholesterol. So the steroid means all those chemicals having this common structural components, then we call them all steroid. That's why do you still remember the cardenolite, that toxin, the monarch butterfly and the milkweed, originally actually the milkweed toxin. Cardenolite also has basically this component as a basic unit, so that's why it can be also called a steroid. And so all these so-called steroid hormone, the reason why they are called steroid is because of that, and all of them are all made, synthesized starting from this cholesterol. And here in this model, especially the space filling model of this molecule, we can easily see why the cholesterol molecule, even it is something like unusual compared with the other more conventional type of lipid molecule, still it should be regarded as a lipid. You can easily see in this molecular model, right? So that's one function, which is important, I know. However, steroid hormone is not something that produced in all of the types of the cells in our body, nor it is something that needed in all other area of in general, all area of the body, different part of the body. Only specific area that steroid hormone can be made, and also only some specific areas are the area that being a target of the steroid hormone. So are there any more general, more essential function of steroid, like cholesterol in our body? Yes, it is actually to the individual general cells aspect. Probably perhaps it is the more important function. It is very essential in maintaining the actual strength of the cell membrane. And another, because of that, also it gets to reduce the permeability of the cell membrane. Both of them are actually related together. So here, this figure shows the presence of this each cholesterol inserted in the alongside with the cell membrane as like in the middle, and having this make the overall the cell membrane less fluid and make more rigid. So the mechanical strength, like I usually take the example of like a building, a concrete building, although it may look so sturdy, formidable outside, but if you don't have any inside the steel beam, then that concrete building will be really very soft, easily breakable. Same thing, just like the cell membrane. Without this inserted, embedded cholesterol molecule, it will be much more feeble, weak, and that's one reason. And another more important reason is, due to this, it will make overall so-called the permeability, how much all those different components these cell membranes are allowing to pass them through can be measured by the different permeability. Overall, that permeability is very important in that overall, like small non-polar molecule like gases, oxygen and carbon dioxide and nitrogen gas, like uncharged and very small, will have extreme easiness of being able to pass through the cell membrane freely. Okay? They don't have any kind of restriction virtually, right? And the second tier of such permeability wise, because it's here, the more you go up, the higher the permeability, and the lower you go down, and then it will become much more difficult to get through this cell membrane. And water is obviously a slightly polar. So even though you may think actually water can be freely passed through the cell membrane, that is not in reality is the case. Water themselves have some kind of a challenging problem to overcome to pass through this cell membrane. And glycerol, which is like a kind of a slightly charged alcohol too. But the bigger challenging problem is this type of a charged. It's not obviously charged but polar molecule, like large uncharged but polar size, and in terms of this polarity, uncharged but still polar due to the presence of a lot of oxygen, and they do have still like a more degree of a difficulty in passing through the cell membrane, and extremely difficulty goes to these small charged ions. Yeah, they have no chance. So at least cells, at least for these two different classes of molecule, which are polar either or charged, which have absolute difficulty in getting through this cell membrane, cell membrane itself should have developed or sophisticated transport system. Otherwise, they will have any difficulty in the efficient traffic, because all these material, even though they are very difficult to pass through, but otherwise they are a very, very essential molecule for the cell's metabolism and as a consequence, survival activity. That's why. So that's why, as a cell membrane, you better have a lot of such different transporter proteins embedded on the cell membrane, which we are going to take a look at later chapter. Okay, so next topic is a nucleic acid. For nucleic acid, although it has a lot more details that we might want to go over, because we are going to go over all those gene structure and the related things in the later chapters. But on the other hand, we don't want to actually have any kind of existing review or preview to some of you about this nucleic acid. So I will try to be very succinct and concise as much as possible. So it's can be characterized as one sentence. You can say nucleic acid as a polymers of nucleotides, which means that sentence alone just reveal what type of monomer is being used in building nucleic acid. Yeah, it is. Its name is nucleotide. Okay. There are two different types of, which is better. Unlike the other categories of macromolecules, we don't really have to worry about different namings, examples. You only have to memorize two, DNA and RNA. These are these two sole members making up the nucleic acid. Okay, so this is the model view of the left side of DNA and the right side is an RNA here. You can easily notice that the DNA is a chubby and RNA is a skinny. The reason is DNA is as you can see in different color coding, DNA is usually exists in a double strand put together with a specific chemical bond. On the other hand, although RNA is also capable of such a thing like a DNA does, but RNA, for some reason, actually there is a good reason why RNA wants to be left alone usually. Usually exists as a single strand. That's the one distinct feature between RNA and DNA. Now, nucleotide. One interesting thing about the nucleotide is, even though it is a monomer from which all this polymeric DNA or RNA is assembled, but nucleotide itself is made up of three different parts. So here are these three different parts. One is the sugar, either ribose sugar. Remember that ribose sugar, 5-carbon sugar, or deoxyribose sugar. We already looked at the difference between ribose and deoxyribose. The only difference was the presence or absence of the oxygen. And actually, the name DNA came from this feature of not having the oxygen at the particular position of that carbon number 2 of the ribose sugar. That's why it is called deoxyribose, and hence the name DNA instead of RNA. RNA stands for ribonucleic acid, ribose. DNA standing for deoxyribonucleic acid. Okay. And so the sugar, and one part is taken up by one of the four different types of so-called nitrogenous base. We usually abbreviate the different types of nitrogenous base part using only single letter notation like AGCT or AGCU, depending on who you are. In the case of AGCT, these are the nucleotide, the bases used in DNA, whereas AGCU, all these four different types of bases are used in making the RNA nucleotide. And whether you are RNA or DNA, the same phosphate group is also taking up one area. So anyway, this sugar and nitrogenous base and phosphate, three parts all together make one component of nucleotide. As you can see over here, nucleotide 1, 5-carbon sugar, nitrogenous base here. And speaking of nitrogenous base, you can have like if you are a DNA, then you can have cytosine, guanine, adenine, and this for either one of these four different types. So in other words, you can have four different types of nucleotide as a DNA nucleotide. Same thing goes for the RNA nucleotide. So if you are a RNA nucleotide, depending on this type, you can have either one of these four different types of nucleotide, depending on what type of nitrogenous base you have. So bottom line is, the RNA and DNA, the thymine, that's why it is abbreviated as a T. It's not. Thymine is replaced with the uracil, so that's why it's usually designated as in U, and vice versa, like the RNA and DNA. That's the only difference as far as this nitrogenous base goes. Of course, there is a difference in the type of a sugar, the carbon sugar. In the case of ribose, the RNA, the ribose sugar is used, and in the case of DNA, deoxyribose sugar is used, as we have seen already. So that's how you can summarize them. And remember the position. Although all these carbons, here actually the carbon is, each point carbon is missing, but instead the carbon is there. Also there is a carbon, and there is a carbon, 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 and carbon, like this. But all these carbons have this some specific number. So around this ribose sugar or deoxyribose sugar, the number 1 carbon goes here, and number 2 carbon, 3, 4, and 5. Okay. These are the conventional, like naming, numbering of these carbons in any organic molecules. So like let's try to have this graph hold this number of carbon, especially on the ribose or deoxyribose, because we are going to have a little small discussion later on when we get to the actual DNA and gene part. So one additional thing is look at this point. Let's take a look at this ATP. This is a structure of an ATP. Base is one particular type of base, and an adenine, we usually abbreviate as an A, like adenine, nitrogen base. And you have a ribose sugar, because you can you notice that okay, still there is an oxygen, so it must be a ribose sugar. And the only difference of this ATP from the other types of such a nucleotide, like RNA making nucleotide, is you have additional 2 more phosphate here, unlike the diagram shown in the previous slide, that having only 1 phosphate in any kind of different nucleotide. However, in reality, actually, all those nucleotides exist in the form of having this 3 phosphate instead of 1, and then by the time they actually has been inserted into the whole chain of DNA or RNA, then only then they actually shed these 2 phosphate off to become this entity. So what's the point here? Like ATP. So ATP is actually the universal version of the energy wherever you go, like in life. It's an ATP as the universal energy source. It is actually a type of nucleotide monomer, right? Of RNA, of course. So in other words, this ATP can be incorporated in forming RNA. And also at the same time, it can have the energy generator, like a rechargeable battery, like a function in all those metabolic reactions. So here comes another versatility of ATP. So the point is, ATP is actually a nucleotide which can be perfectly okay to be incorporated into the growing chain of RNA. And in DNA especially, because RNA we don't really hear, although it still has a equal capability of this complementary base pairing that we are going to take a look at. But let's take a moment to look at the future this complementary base pairing in DNA only for now. This complementary base pairing means there is a specific hydrogen bond between 2 nitrogenous bases, in particular between A and T and G and C. Notice that between A and T there's a 2, which means double bond, and there are 3 lines, triple. But even though I just put this in solid line, that doesn't mean that they are a covalent bond. All of them are like in nature they are hydrogen bond. So as you can see over here, between G, guanine, and C, cytosine, you have all these hydrogen bonds. 3 hydrogen bonds are formed naturally. So these are the force that hold the 2 different nucleotides together, and overall this is the force making possible the existence of the DNA as a DNA double helical structure, DNA double strand. Same thing. So between A, adenine versus thymine, 2 nitrogenous bases, only 2 hydrogen bond, and that's where this so-called specificity of this so-called the complementary base pair comes, because the number of hydrogen bonds that can be formed between A and T and separately G and C is different. That's why it's just like the plug, like a Korean plug, you have these 3 plug things, like electric cord, power cord, and if you go to the US, you have this 2 plug thing. So those electrical devices won't fit without any adapter. So similarly in here, because of these 2 intrinsic difference between the number of this plug, A only will be able to normally. Okay. If we go deeper, that's a different story, but I don't want to reveal that. Let's just put that under the bed. So only A and T can form this specific hydrogen bond, whereas G and C at the same time have such a specific hydrogen bond only. That's what it makes this so-called specificity of a pairing we by now recognize as a so-called putting the name of complementary base pairing. It's only between A and T and the G and C. Okay? So
Nucleic acids are polymers formed of monomers called nucleotides. DNA's two strands wind around each other to form a double helix, with hydrogen bonds forming between the nucleotides on opposing strands. RNA is a single-stranded molecule that can form hydrogen bonds between nucleotides in segments of itself. RNA is thus capable of great structural complexity. Nucleotides have three components: a phosphate group, a sugar, and a base. In RNA, the sugar is ribose. In DNA, the sugar is deoxyribose, which is ribose with an oxygen atom removed, a small chemical difference that actually accounts for the quite different roles of DNA and RNA. There are 4 different bases in DNA, abbreviated A, T, C, and G. A forms pairs with T and G with C. RNA has an altered form of T called U, which forms pairs with A, just like T in DNA. DNA is the storage form of hereditary information. RNA is formed by copying the nucleotide sequence in DNA. RNA carries the message in DNA to other parts of the cell and can also fill structural roles.
Okay, so this I just before this short movie, as I just mentioned, this complementary base pairing is the one, the force, responsible for holding these 2 DNA strands together, like this. Okay. And as I mentioned, the only reason why they have this twisted like a helical structure is because this chain is too long. Okay. And hydrogen bond between these. And why these 2 opposite strands are like arranged in upside down, we will worry those things later, not this time. Okay, so for now, just postpone the worry. This complementary base pairing is actually the significance is this is the one that made this DNA to be able to function as the genetic material. What I mean by that is this, because this complementary base pairing, when the new DNA is trying to be synthesized, right, and then it's actually become very, very clear and straightforward. That I usually take the example of having a mirror. When you try to make up, one of the most essential tool is the mirror, so that by looking at this mirror, you can have any adjustment. Same thing goes for this DNA. When DNA is trying to replicate, because of this complementary base pairing feature, you know by itself, you know what type of the nucleotide will come to synthesize. So these green ribbons represent the newly synthesized strand during the DNA replication. So to do this, you don't really need any kind of a special template out of which the new DNA synthesis is based upon. Your existing cell can serve as a template for new DNA to be synthesized. This is such an extremely important feature of the DNA to be able to function as the genetic material is the thing that I need to emphasize. Okay. Now, the last class of the biomacromolecule, the proteins. Similarly, we can define the protein just like we did in the case of the the nucleic acid. What we can say about protein is the polymers of amino acid. By doing so, we can identify what is the monomeric form of these all polymer proteins. So the amino acids are the monomeric components with which all these different types of proteins are built. So the type of condensation, the covalent bond, the condensation reaction used in protein, its name is peptide bond. So rather than usually in that I just specifically mention, not only mentioned but also write the name of the peptide bond is because another name for calling protein is polypeptide. Sometimes proteins are called polypeptides, and actually it bears such chemical bond, peptide bond, and that's why the name polypeptides was formulated. And another reason I specifically also brought up the attention to this peptide bond is, unlike other types of all the condensation bond occurring in the nucleic acid and the carbohydrates and lipids, this peptide bond formation only takes place only in one place in the cell, which is on the ribosome. So unlike others that can happen in anywhere in the cell, the peptide bond formation can only occur in the ribosome, which is actually catalyzed by, surprisingly, we will just get to that later, very surprising molecules. So this is the standard basic structure of any amino acid, which is a little bit of unnecessary for non-science major to worry about this structure, but simply to show some basic standard architecture and some additional part which makes each different amino acid unique. And actually that's the reason why protein can generate so many different type of shape. So as a consequence, protein's function is, like as we went over, specialty. Proteins are very special in their structures and as a consequence the functions. So here one part, so the left arm is occupied by a specific chemical group called amine group. So it is an amine or amino group. So amino group. And here another part, opposite side, is occupied by another chemical group we identify as a carboxyl group. Okay. So that's the standard. Every different amino acid, like unlike the case of nucleic acid, where only 4 different types of nucleotides exist in the cell, here in protein building job, 20 different amino acids are available. Actually, the real thing is there are more than 20, it's probably hundreds different amino acids theoretically can exist. And actually more than 20, obviously many more amino acids than mere 20 actually exist in the cell, but for some reason there are only this particular 20 different types of amino acids are the ones participating in the protein synthesis process. So regardless of any different types of amino acid, all of them must have this same component structure of at the center you have to have one carbon, and on the other both sides one amino group and other carboxyl group, and there is one hydrogen. Remember, carbon, as a one single carbon, you have a 4 capacity of having covalent bond around it. And the remaining last position here labeled as an R group, because R can be anything, like even hydrogen can become an R. So here is the area where actually many different types of chemical group can be attached, so that overall make this particular amino acids very unique amino acids. So that's the secret of having many different types of amino acid. Other than this part we label as an R group, or other remaining part as a basic backbone so to speak, this area is identical to any different types of amino acid, and usually this amino group is in and natural we describe it as a physiological condition, natural condition in the cell. Amino group is usually take a positive charge. Because of this positive charge means this amino group has a slight tendency of acting as a alkali, a base, and the carboxyl group acting as an acid. Acid means it has a strong tendency to dump its hydrogen ion, and once afterwards this part will become negatively charged at a neutral pH, if you still remember what pH stands for. So this is usually each individual amino acid's status, having one side is a slightly positively charged and the other side is slightly negatively charged. Altogether it will balance out all those charge differentials. However, individual part, obviously still they are kind of a polar charged area. And another thing is this, here in an R group position, you can have many different types of properties of chemical can come. So depending on different types of amino acid, some amino acid can become highly polar, some amino acid can become highly non-polar. So that's also what makes the whole different properties for each different type of amino acid, right?
Proteins are polymers consisting of strings of amino acids. Amino acids are small molecules containing a central carbon with 4 groups attached: a nitrogen-containing amino group, an oxygen-containing group, a hydrogen atom, and a side chain. There are 20 different amino acids, identified by their different side chains. When proteins are synthesized in cells, they immediately fold up into their active 3-dimensional conformation. The final folded shape is determined by the sequence of amino acids in the peptide chain. The shapes of folded proteins are stabilized by internal hydrogen bonds and ionic bonds. Proteins are both flexible and durable. In cells, proteins last anywhere from a few hours to several days before they are digested into free amino acids, which are recycled to create new proteins.
So here, one of the important point that I'd like to address once again is in finalizing the particular structure of a protein, several different chemical bonds are utilized, as this video mentioned, the hydrogen bond and then ionic interactions, but that's not all. So overall, that the types of chemical bonds that are important in determining the final protein structure is all very weak chemical bonds. So in other words, the covalent bond is not something utilized in protein structure determination. There is one particular exception, but particular type of covalent bond can be used, but other than that, almost exclusively not a covalent. So weak non-covalent bond are the one, just like the example we have seen in the case of the antibody and antigen interaction, which mainly the van der Waals interaction is acting as a major role, and DNA double helical structure, the maintenance is maintained actually hydrogen bond like that, and DNA and protein interactions are usually maintained through ionic bond and some other type of weak chemical bond. Overall protein structure is maintained by many weak non-covalent bond, including the hydrogen bond and ionic, also van der Waals, all, and among this also a very important chemical interaction that has a really important determinant is the hydrophobic. This hydrophobic interaction, where because all this environment is an aqueous environment, a watery environment, so the hydrophobic part, so depending on the nature of the chemical nature of different amino acids, some area could be a hydrophobic area, some area could be a hydrophilic area, because of the many different types of R groups can attach to the last position of that base of amino acid, carbon. So because of that, hydrophobic area will aggregate together, will fold inside to avoid any exposure of themselves to the external watery environment. And that's the actually one primary driving force to determine the final protein structure. So once those hydrophobic area fold inside, and then the surface area will be taken up by those area which can handle those polar watery environment. So that is very important. The force, driving force of a protein structure. So as been already mentioned, the 20 different amino acids on the ribosome, actually, using 20 different amino acids assembled together, like this. Well, I found this cartoon on the internet, and I love this cartoon because it has a really neat resolution. So someday, if I had enough time, then I will try to copy this drawing exactly by myself. I love to do such things. But anyway, here what you can identify what's going on over here is here these two areas are like two different subunits of the ribosome. Well, we are going to take a look at this like later in probably chapter 5 or chapter 4 when we get to deal with the DNA and gene expression saga. So anyway, the ribosomes are made up of two parts. Both of them are actually the mixture of different types of RNA and the different types of proteins. And this is called the large subunit and the small subunit. And being sandwiched by these two subunits is an RNA, messenger RNA. This messenger RNA is just a copy of your DNA information, which we are going to take a look at, and then find out, have the exact relationship in the later chapter, like chapter 5. But the thing is, so the point is, what type of amino acid, the order of amino acid will be determined by the gene in the form of this, the information carried by this messenger RNA, and according to that messenger RNA, specific different amino acids can be brought up to this particular location where amino acid, each amino acid is being carried by specific type of RNA called tRNA, standing for transfer RNA. So they are like by transferring one amino acid into the other. So like this is a growing chain of the amino acid. This is basically what's happening in the amino acid synthesis. Actually, this is a much more accurate description of this in the in picture-wise description. So existing this growing polypeptide, the chain of a polypeptide, will always be transferred into the next newly coming transfer RNA carrying a new amino acid, so that transfer entire amino acid chain into the new guy, and then having one more growth in terms of this number of amino acids. That's how you do this process continue until certain time point when it reaches to the end point, and then this synthesized protein as a chain of amino acid is released, like that. So like if this whole description, the picture is a little bit of confusing or intimidating for you at this point, you don't have to worry anything about this. It's just we will be able to see more of this figure later. So at this point, just because everything is so new to you, doesn't have to make you worry. And another thing is, usually along with this particular messenger RNA, it's given time, not only one but many multiples of such the ribosomes are traveling, moving through this one particular messenger RNA, so that you can generate multiple copies of the protein, complete this polypeptide, which is equal to protein, can be generated at the same time using this one template of this messenger RNA for this protein synthesis is showing in this diagram. Okay. So what's most important about the overall description of this protein is this. So due to the nature of different types of amino acid that can be randomly arranged, actually we shouldn't say randomly arranged, but according to the different DNA sequence that is responsible for making this particular protein, so you can generate so many diverse form of such order, arrangement of or combination of the amino acids, so that you can generate so many diverse shapes of protein, which means if you have a diverse shape, then you can have diverse functions, because always shape dictates the function. Like, so what kind of other functions? Like you name it, like enzymes, yeah. Why carbohydrates and lipids and DNAs and RNA, a little bit of exceptions, cannot usually make a good enzymes, but exclusively proteins. Why only proteins are heavily utilized to function as enzymes? Is the reason is there because of this diverse shape that can be generated by protein. Other macromolecules cannot have such ability, with the exception of RNA. RNA somehow can, but we will see an example because of the RNA structure later in next chapter. And receptors, you know what the receptors are, and transporters, right? And antibodies, a structural protein, you name it. There's so many different types of function can come from proteins, like enzyme, antibody, hormone. All these different structures are shown in different types of model, beautiful model, structural model. So an example of like a more friendly way of showing the different function of the proteins are here. Okay. And also structure of protein, like feathers and spider web and hairs of insect and your the fingernails and your hairs too. All these are the example of such example of structural proteins too.
Enzymes are proteins that carry out chemical reactions. They have two special features. First, they are highly specific. Each enzyme acts only on select molecules called its substrates. Second, enzymes are catalysts. They greatly accelerate the rate of reactions they carry out, and they are unchanged by the reactions they carry out. Enzymes don't get used up by chemical reactions. They carry out the same reaction over and over as long as their substrate is available. Enzymes have a recessed pocket on their surface called the active site. The shape of this pocket precisely matches the shape of the substrate. Molecules that don't bind snugly in the active site don't react. The shape of the binding pocket is the key to enzyme specificity. This enzyme is chymotrypsin, which breaks down protein molecules in the stomach during digestion. Note how precisely the large side chain of a specific amino acid fits in the depression within the binding site. That precise fit positions specific atoms of the substrate for reaction with atoms from the enzyme. Enzymes work by lowering the activation energy of a reaction. Biological molecules are generally very stable. They don't spontaneously break down because the activation energy for that reaction is too high. Enzymes lower the activation energy by positioning the substrate in just the right shape at just the right distance from a reaction partner for the reaction to occur in the active site. When the reaction is completed, the products diffuse out of the enzyme. You might wonder why the products don't stick in the active site, since the substrate fits so well. It is because the products are now different molecules with bonds and electrons in different locations. The active site binds the substrate with just enough energy to promote the reaction, but the product diffuses freely away. When the reaction is completed, the enzyme is exactly as it was before the reaction, ready to react again.
Okay, with that, now we can conclude the all discussions about the chemistry of life, the culminating in the protein structure and how the enzyme acts, the mechanism of the enzyme action. So although my original intention was not really this much intensive discussion going over chemistry of life, but once getting started, then several many important aspects, although I try to explain as simple as possible as I can do, but some of the important points I cannot really pass up. That's how this whole lecture series can become a 2 weeks worth lecture series of relatively existing one, but hopefully we at least can have a clear definition and an idea and understanding of all these different chemical bonds, and with that resulting in many different biologically important macromolecules. Okay, having said that, so once again wishing you having the nice weekend. So see you until next week.
Never started saving money, but everything is different since you've been around. It's the way you're smiling at me, it's in the way you hold my hand. It's the way I've watched you change me from a boy into a man. It's a million things about you, and I don't know what it is, but I have never known a love like this. Never used to get excited to sit here in the silence, holding on to something the way I'm holding you. Didn't used to know how fast time walks and runs and flies by. Never thought I'd feel so deeply, but damn I do. It's the way you're smiling at me, it's in the way you hold my hand. I've watched you change me from a boy into a man. It's a million things about you, and I don't know what it is, but I've never known a love like this. It's funny how everything I dreamed about starts to seem so empty without you. It's the way you're smiling at me, it's in the way you hold my hand. It's the way I've watched you change me from a boy into a man. It's a million things about you, and I don't know